I'm waiting for you, Rick.
I'm hitting the button.
All right, excellent. Well, welcome everybody. We are very glad that you are here with us today. My name is Ann Merchant and I work for the National Academy of Sciences. And now I am joined by
Rick Levert. I'm the program director of the Science and Entertainment Exchange, which is also a program of the National Academy of Sciences. Sorry about that. My video button was sticking on the
It can
thing.
be elusive. I know how that feels. It's like we have to do this sort of like get the mute button, get the screen share stop, do all the stuff. We're not always that good. That's all right. We try. But I have to say, it's really great to see your background now that you do have that on, because that is the Great Hall at the National Academy of Sciences, adorned for TED at NAS, which took place on November 1st, 2019. It was a great day. And we were really proud to see our building. It always looks pretty great, but it was pretty spiffy on that particular day. So it's nice to see that on your background. I'm glad you did that. I'm still in my same corner. I have nothing special going on.
Thousands of miles away from the NAS.
Yeah, exactly. Exactly. So, and as always, we will start in our customary way, which of course is to at least talk a little bit about the National Academy of Sciences, started in 1863 by Abraham Lincoln. We always like when we get to do slide decks about the Academy, because we get to pick some great photos and things from our building that sort of feature. We have a wonderful painting that features Abraham Lincoln in our boardroom, which I understand is sort of like an early version of Photoshop, because he didn't actually sit for the portrait. So they painted in everybody else and then painted in Abraham Lincoln afterward. But we always like to there it is, there's the portrait. So we like to lay claim to Abraham Lincoln as being the president who signed the National Academy of Sciences into being through executive order. And of course, all these years later, here we are doing exactly what President Lincoln imagined that we would do, which is help the nation steer a good course towards science policy that made sense and was evidence-based, and that was based on the input of experts. And of course, conceived originally as an honorific society, we still do have members of the National Academy of Sciences. It's a rarefied group, one of whom will be on our virtual stage today. And then, of course, over time, we were joined by the National Academy of Engineering and the National Academy of Medicine, which are also honorific societies, but together they formed the National Academies of Sciences, Engineering, and Medicine. And one of the things that, of course, that I did want to say is that people who are sort of our core, hardcore constituency audiences, they're very used to the big 400-page reports that we put out that are part of steering that good course towards science policy that makes sense. But we've also been doing something new called Rapid Expert Consultations, or as we like to call them, REX, because we have to make an acronym out of everything. But they've been a really interesting way that we've responded during the pandemic to the need for good expert input, is that we haven't been doing these, we don't have time for a 2-year study, that sometimes science input is needed very quickly and in an acute situation. So we have a new one that came out this past week, and it's on disaster planning during COVID. So that we saw in Texas, for example, we're asking people to leave their homes and shelter in a mass evacuation site. And what are the concerns if you're also being told to socially distance and not be in places where lots of folks are gathering? And so how do we balance those considerations? Which is more dangerous? And so they've looked more carefully at how we might respond to future disasters, because we can't expect that there won't be more of them, and disasters don't stop just because we're dealing with a pandemic. So I think Sachi has put in the chat a little link, and we urge you to go look at some of those rapid expert consultations. They're super short and very interesting, and oftentimes very useful. But of course, the program like the Science and Entertainment Exchange also exists to be able to communicate more broadly about our work in very different forms. And I will leave it to you, Rick, to explain that.
Yeah, so if you're a storyteller in a mass media format, writer, producer, executive, you make films, TV shows, video games, novels, you have a question about science, you can call us at 844-NEED-SCI, or you can send us an email, and Sachi's putting our contact information up for you. And we will connect you with someone who is a field expert who can answer your question as it relates to science, technology, engineering, and medicine. We've done over 300 events like this one. We've done over 3,300 consults on documentary films and feature films, including Avengers Infinity War, Man of Steel, Black Panther, Captain Marvel, more recently Songbird. And if you're a storyteller, please reach out to us. If you're a STEM professional, you're just hearing about what we do, definitely we've had lots of people who've said, hey, I saw you in an online event and I'm interested in participating. And we're always looking for more scientists and experts. I want to quickly thank our AV wizard, Ameche, for all of the back end stuff that you do. I want to thank the office in DC, Courtney Sloan, Jeff Fishman, and Sachi in Los Angeles. We couldn't, these events take a lot of back end stuff. So thank you, everybody. Our sponsors, Howard Hughes Medical Institute, who's responsible for this event today specifically, and all of our online events. We couldn't do them without you. Also, our sponsors, the Alfred P. Sloan Foundation, Walt Disney Company, Film Nation, Google, the Lyda Hill Foundation, Corteva, and individual donors like so many of you. We've got our 2021 challenge coins coming very, very soon. They're supposed to be here in a week or two, according to the people who, strike them. So get those out to you for everybody who joined at the supporter level. You saw the slide, hopefully at the beginning of the show. Thank you so much for supporting us in that way. It goes right back into making more of these events. So thank you for your generosity. Today's event is going to be moderated by, a real professional instead of, one of us or me. So I'm very excited to have David Biello here. I will be in the background feeding him your questions. So if at any point you have a question, you can click the little box here or click the little box here, depending on what format you're using. And we will be feeding questions to David. There will be many more questions than we have time to answer. If we don't get to your question, it is my fault, not David's. And so, please don't hesitate to ask a question if you have one at any point today.
That's right. Before we hand it over to you, David, we have made it a weekly habit to do something that we call our rabbit hole. And for me, I discovered something called Radio Garden. I didn't know about this, but it was created by it was a Dutch project from the Netherlands Institute for Sound and Vision. And basically, you can sort of spin the globe and find live radio from all over the world and listen in. I was listening to Train being played on a Czech Republic station, country music being played in Wagram, Austria. There was sea chanties coming out of Ireland. And then I was listening to, it sounded like an American Christian radio station, but it was coming out of Wakari, Nigeria. So it's super interesting. Sachi put the link in the chat and you can spend hours getting lost in it. It's really fun. I highly recommend it.
Does it make you feel like you were there, Anne?
Well, yeah, I mean, in some ways it was weird because I felt like I was still here. Like I didn't expect to hear country music in Austria, but that's okay.
So I want to recommend two things for my rabbit hole. The first one is the Marvel show WandaVision. Really enjoying that show on Disney Plus. So it's getting more interesting with each episode. It's a little bit of a slow burn. And then the other thing, a little bit selfishly, because we are going to have this gentleman on our stage on March 31st. If you want to get ahead of the curve and check out Kim Stanley Robinson's new book, The Ministry for the Future, I'm reading it this week and enjoying it greatly. So, and as I said, if you have questions for Kim Stanley Robinson about a month from now, you'll get a chance to ask them on this stage.
Yep. And I'm coming right behind you because I have the book on order myself. So you don't give it away. No spoilers. All right, good. So, David, we are going to hand it over to you, but I did want to say that, how much the Academy, and of course, we were joined by amazing sponsors from the Simons Foundation and the Kavli Foundation who made TED at NAS possible. This was an event that I think, we had said it was years in the making, and it really was. It was lots of conversations with the TED organization, and of course, among the three partners with the National Academy of Sciences, Kavli and Simons. And it was just, I think it was a real labor, but a labor of love. And we were also proud of the event that actually graced the Academy's stage on that date. But, you know, we couldn't have done it without TED. You know, it was the first ever science lineup for TED, but it was, you know, as we kept saying, but it's real TED, it's real TED. And we don't want to marginalize the TEDx events, but, you know, we depended on people like you, David, and the other professionals at TED to lead us through this process and to ensure that this was a successful event. And so we're grateful to you from that day and onward for being here and doing this, because you're the TED Science guy. And so we wouldn't want to do this without you either. So we're really happy that you're here, and we're going to hand it over to you and our 3 reprise speakers from TED and NAS, because they're all pretty awesome too. So we're going to let you take it away.
Well, thank you, Anne and Rick, and I will keep the cavalcade of thanks going by saying it was an incredible honor and privilege to work with the both of you and all of our partners at NAS on the TED at NAS event. For those of you who tuned in early, you saw some of the pictures from the event. That was like a glimpse into the before times. I saw a crowded room of people without masks. It was somewhat upsetting, somewhat wistful, and brought back many fond memories. So my hope here today is to bring you, I guess, some great ideas to inspire, I don't know, either your science or your entertainment, assuming that you are in one of those 2 fields. As Anne mentioned, I am David Biello, and I am the science curator for TED. What does that mean? That means I pick the scientists and work with them to give a TED Talk. Which can be a fraught process. And if you want to know more about that, you probably want to ask the people I'm about to introduce. I'm not going to talk a lot about TED, as I suspect that many of you may have seen a TED Talk, and I'll just leave it to your questions to get into anything you want to know about TED. What I will say is that I can't teach you anything that the ancient Greeks, who were amazing rhetoricians, wouldn't have been able to teach you. I will, however, talk about NAS's, I will, however, talk about TED at NAS, which was TED's, as Anne mentioned, very first science-only event. By which I mean, we didn't only have science speakers, but even the art had a science theme to it. In this case, a musical act and some poetry, which you should go back and check out. It was a truly incredible event, and you've got 3 of the participants here. Like I said, if you want to know what I really do, you should ask them. And I should also know that you should check out all the NAS talks, because they really are amazing, not just the 3 that we have represented here. We had everything from sex chromosomes and axolotl expert nuns to teachings about how to stay curious, and also the definition of a stellar mortician, which I think sounds like an interesting character. And he happens to know everything about where you came from. So definitely check those out. And I hope you have already checked out our speakers here today. I hope you've checked out their talks. Starting us off with a bang is the one and only Bethany Edwards, an oceanographer and a professor at Berkeley. She gave an absolutely amazing talk about mastering the chemical language inside of you through the cells living in the ocean. And she's going to update us on the latest and greatest from the most significant feature of the planet behind me. Take it away, Bethany.
Thank you so much, David. Let's get my slides on the screen. There we are. So I left you at the end of my TED Talk with this idea about this chemical language, this chemical signaling language based on oxylipin compounds, and how it coordinates this intricate dance between the cells of your immune system to respond to wounding, viral infection, bacterial infection, and how it also coordinates this really intricate dance between different cell types of microbes in the ocean. And today I'm going to tell you about how oxylipins, there we go. So today I'm going to tell you about how oxylipins and this chemical signaling language between marine microbes impacts carbon sequestration in the ocean. So how much carbon is actually stored in the ocean. So since the Industrial Revolution, since we started burning fossil fuels, the oceans absorbed about 40% of the carbon that we've liberated through fossil fuel burning. And this is due in part to the ocean's biology, specifically phytoplankton, which are like tiny plants, and they take up CO2 through photosynthesis, and they sequester it in the ocean. Phytoplankton are the beginning of the marine food web. And so as humans with a taste for seafood, we might be more interested in the carbon that's transferred up the food web from phytoplankton to fish. But actually, the majority of the carbon that phytoplankton photosynthesize is cycled through microbial communities, which are really complex. And so my lab at Berkeley, what our goal is to untangle these interactions so that we can better understand the fate of all of this carbon. And because these microbial communities are so complicated, there's actually a lot of uncertainty about how much carbon is stored in the ocean, how long it's stored there, and how can we predict this in climate models. So some of the questions that my lab asks includes, how efficiently is carbon stored in the ocean if phytoplankton die, aggregate together, and sink to the bottom, versus if they're eaten by tiny zooplankton, like these microzooplankton here? And is that different than if they're eaten by larger zooplankton? Or what happens to the carbon if they're infected by viruses? We also ask questions about bacteria that live off of this dead phytoplankton. What controls how quickly these bacteria turn these phytoplankton back, decompose them back into CO2 and nutrients? And how do these processes vary in time and space? So from year to year, over the season, over different ecosystems. And then finally, can we lay oxylipins? Can we lay this chemical signaling language on top of all of this? And kind of the holy grail for microbial oceanographers is to be able to see something in the environment and then get those organisms, grow them up in the lab, and see if we can, tease apart these questions. So I'm going to show you one example where we've observed oxylipin being spoken in the ocean, and then we've gone back into the lab to try to understand it better. So in 2013, I went on a research cruise along the coast of California. Here you're looking at a satellite image of chlorophyll over the 8 days that we were there. So you can actually see phytoplankton from space. And each of these black dots is somewhere where we stopped to sample. And you can, we use genetic techniques to look at the phytoplankton community. And we saw that the phytoplankton community shifted between along our cruise track. So you can see Monterey Bay has a very different phytoplankton community compared to closer to San Francisco Bay off Point Reyes. And we also looked at the viral community that was present in this water using the same genetic techniques. And so we looked at free-living viruses, so viruses that have already killed their host and liberated themselves. And we compared that to viruses that were still inside of cells. And so here we're looking at values above 1 are showing you that you're later in infection, and values lower than 1 show you that we're in the early stages of infection, where the viruses are still replicating themselves inside of their hosts. And so you can see that the phytoplankton community at Point Reyes was kind of in this post-infection state, where you've already had a lot of viruses liberated into the water, whereas Monterey Bay was a little bit earlier infection, or active infection, where you have more viruses inside of the cells. And when we looked at the oxylipins across this cruise transect, we see that there's more oxylipins associated with this post-infection state at Point Reyes. So what you're looking at here is the relative abundance of every single oxylipin compound that we observed in these water samples in each of our sampling sites. And so red values show an increase in these compounds compared to the other samples. And you see that these post-infection sites associated with point rays have a lot more oxylipins in the water column. So this is cool. We've observed this chemical signaling language in the water column. But how are these oxylipins impacting carbon cycling? And so if we took a closer look at exactly what compounds are being produced, and we found that there were several compounds that we know are bioactive on members of the microbial community. So this is one example. So this is a compound, dihydroxycosapentanoic acid, and it was much higher in our post-infection sites. If you've ever taken fish oil, which has fatty acids EPA and DHA, this is basically EPA with 2 hydroxy groups added to it. We see here in pink, it's much higher in those post-infection sites near point rays. And we set up some experiments where we actually infected diatoms, these phytoplankton, in the lab with viruses. And we saw that we got the production of this compound. So this kind of confirms for us that seeing it in the environment, in this place where we know viral infection has just happened, and then seeing it in the lab where we've infected phytoplankton with viruses, we're pretty confident that that's the signal we're looking at. We also set up experiments where we added this compound back in to some of these little microzooplankton grazers, and then also to some bacterial bacteria that we collected from the environment. And we saw that these compounds inhibit the grazing of these little micro grazers. Some other people found that these compounds decrease the reproductive success of larger grazers. And we also saw that these compounds stimulated the bacteria that were living off of this dead phytoplankton material, and increased the amount of CO2 that was being recycled back into the atmosphere. So we think that these oxylipins might actually be decreasing carbon sequestration in the ocean by stimulating these bacteria. And so kind of the next steps for us are to monitor how much carbon is being stored. And so hopefully NSF will bite on this idea. If you have any rich friends or want to drop 100 racks on some sediment traps and support science, our plan is to go out here to the San Pedro Ocean Time Series, which sits between the Port of Los Angeles and Catalina Island, and to measure carbon flux, so dead phytoplankton sinking out of the surface ocean over the year, and to monitor the community and oxylipins to see if, when we see viral infection, do we see less carbon export than what we might expect? I'd like to thank all the people who helped to bring this science to you today. And I'm happy to answer questions at the end. And I will hand it back to David.
Thank you, Bethany.
Thank you.
You also win the award for pronunciation for saying that long chemical phrase. So kudos to you. Now on with the show. So from the depths of the ocean, we move on to the depths of space. Next up is Mike Brown, who's probably best known as the man who killed Pluto, the planet, that is. He's also an astronomer at Caltech, but he may well be best known in future for discovering a new Planet 9. So tell us what's the latest and greatest from that search. Mike, take it away.
Thanks, David. By now, I hope you've at least had a chance to hear the story of Planet 9, how the most distant known objects in our solar system seem to be tugged in their orbits by a giant unseen planet far beyond any of the planets that we currently know of. And for you careful historians of science out there, you may recall that this is something like the 35th time since 1846 where someone claimed that they could detect the effects of a distant unseen planet. You might also recall that every single previous time someone made that claim, they were subsequently proven wrong. So it probably sounds a little crazy to say, look, I know those first 34 were all wrong, but we are correct about this. But I'm here to tell you those first 34 were wrong, and we're correct about this. And I'll talk about that a little bit more at the end. But rather than going into the details of Planet 9, yet again. I actually think it's really interesting to look at stories from the not very distant past of announcements of potentially controversial scientific hypotheses, discoveries, and then the reaction of the scientific community to really understand how these things work and what you should be hearing from the scientific community right now. So I want to take you back on 4 different stories. First one takes back a while, but some of you may remember this. 30 years ago, there was a claim of something called cold fusion. Raise your hand if you remember. I remember. Everybody, yeah. I see a couple of you raise your hands. Cold fusion. David remembers. Thank you, David. There were these 2 chemists from Utah. I think they were in Utah, and they claimed that they had basically done fusion reactions, desktop fusion reactions with no added heat. Fusion reactions. Fusion reactions are these things that happen in the interior of the sun at ridiculous temperatures and ridiculous pressures, or hydrogen bombs, also ridiculous temperatures and pressures. If you could do fusion inside of a little desktop thing with no extra energy added, you would essentially have free energy to power the world for life, free, clean energy. That'd be pretty awesome. It was a bit of a strange claim, and physicists were kind of astounded and skeptical. It was strange because nobody really had any idea how it could possibly work. And also it didn't really, even the chemists who proposed it didn't have sort of a clear picture, like it does this, but it doesn't do this. They're like, well, it kind of does this and it kind of does that. So physicists and chemists spent some time trying to reproduce the results, and nobody could really reproduce the results. Nobody could make that cold fusion on a desktop work. And the idea was, it rattled around for a couple of years, but it was eventually pretty quickly discarded. The original chemists who proposed this never backed down. And they continue to argue that this cold fusion is real if you just do it right. You have to do it the way they do it, or it doesn't count. And generally, that gets you labeled a crank by other scientists in the field. So that was one idea that didn't work so well. Another interesting example, more recent, not that more recent, 20 years ago now, is the idea of dark energy that I'm sure many of you have heard about. Dark energy is this idea that the vacuum of the universe contains energy that pushes it apart and makes the vacuum bigger. If you don't understand what that means, then join every other person on the planet. I think I have no idea what that means. I mean, I can write down the equations and I understand the implications physically, but I don't know what that means. But somehow the vacuum has energy that expands. Seems a little bit crazy. So this first came up, really the idea of it first came up, or at least the observations suggesting it was true, first came up about 20 years ago when some astronomers showed, or tried to show, thought they showed, that distant galaxies were not only expanding, we all know that the universe is expanding, but the universe was not just expanding, but that expansion was getting faster because of this dark energy. So they wrote this paper saying there's dark energy, and astronomers and physicists just thought, I mean, it's kind of crazy sounding because it's kind of crazy sounding. So astronomers went over the original data with fine-tooth comb, and they suggested, yeah, maybe there's something fishy with the objects that were observed. The original astronomers made some subtle errors in their calculations. Maybe the techniques were not quite good enough to detect such a small effect. If you were paying attention back then, 20 years ago when this first came out, you would see new scientific papers come up every month or so explaining why dark energy was not real, what mistake had been made. And these arguments continued until a new set of measurements came along that agreed with the predictions from the old set of measurements and showed the same thing. And then people tried to tear down those new measurements. And then there were new measurements, and they agreed too, and there were new different predictions, and all of those agreed. And by now, pretty much everyone agrees something like dark energy is out there. And those original people with that original totally crazy idea that a lot of people didn't believe now have Nobel Prizes in their pocket. Maybe they're around their necks. So around their necks as they walk around. I'm sure they wear them all the time. I'm going to give you 2 more very recent examples because I love these. And you might remember these because the first one was just from last year. You probably remember this. There was a claim from last year of the detection of phosphine in the atmosphere of Venus. phosphine is a weird chemical that is on the Earth. The only place that phosphine is made is in biological systems, in microbes, in people, and things like that. So the claim was that phosphine in the atmosphere of Venus was evidence that there are microbes living in the clouds of Venus. Okay, it sounds crazy because it sounds crazy. I mean, most astronomers were pretty skeptical, again, because the argument is basically, we see phosphine, we can't come up with any other reason for it to be there. Microbes on Earth make phosphine, so there must be microbes living in the clouds of Venus, which, as arguments go, I would say that's a pretty desperate stretch. I think this one is going to end up going away on the basis of the fact that other astronomers looking at the data have concluded that there's no phosphine in the atmosphere of Venus. The original people who proposed this have reanalyzed the data and said, okay, you're right, our original analysis was not quite right, and there's still phosphine, but it's 10 times less than we said it was to begin with. That's a pretty bad sign when the direction of the strength of your argument starts to go down like that. Your argument is probably going to eventually go away. I think these ideas are going to fade, and even if there were phosphine, which it sounds like they're not, connecting it to, therefore there must be microbes in the clouds of Venus seems pretty out there. I'm going to give you the last example because this one has been in the news recently, and I'm going to try to win the pronunciations prize at the same time. A couple years back, you will all remember maybe that this object from outside our solar system came screaming through the solar system. First time we'd ever seen a large object from clearly not our own solar system passing through our solar system. This object was given the Hawaiian name Oumuamua. I can pronounce it, but I probably couldn't spell it for you, but it starts with an apostrophe. And there were some weird things about it. It appeared to be very elongated compared to things that we see on in our own skies, like in asteroids. It didn't look like a comet. It was weird, and then it moved a little bit weird. And so, there were rightly some interesting ideas on what exactly it was and how it got there. And then there was this idea, I'm sure you've heard about in the news recently, that it must have been, of course, an alien spaceship. And the reason it moved strangely because it was an alien spaceship with a solar sail, and that's what we were seeing with the weird shape. And the solar sail got caught by the sun and moved a little bit strangely. So as you might guess, that's not a very popular opinion amongst all but approximately one astronomers out there. And the argument is, well, there's weird things going on and I can't think of any other ideas, therefore it must be aliens, which again is not really a very strong argument. And where this argument, I think, goes the worst is that when people disagree with the argument, the proponent of it mostly just argues that science is done incorrectly and we should all do science differently. It's pretty bad when you have to start to make those arguments that the reason that no one believes you is because all science is done wrong. So that one is going to go away, but it's the original proponent will probably never back down, and is generally considered by many in the astronomical community the way one-time well-respected physicist astronomer out there, kind of gone off the deep end a little bit. So with these 4 examples, 3 of them that probably not going to work out so well, one which worked out spectacularly. There are a couple things to take home. One is that most kind of crazy out there ideas that you hear, they're probably crazy and they're probably out there. And 2 is that there's some characteristics of ideas that are going to actually turn out to be okay that we should look into. So the interesting question is, where is Planet 9 in this story? Where does the Planet 9 hypothesis sit? I would argue, not surprisingly, that we're closer to that second case of dark energy, sadly, minus the eventual Nobel Prize, because you don't get Nobel Prizes for discovering planets. I think we're closer to that case than to the other 3. But really, how would you, as just some outsider listening to scientists talk about these things, how would you know? Because we're at that stage now where everyone is trying to pick apart the data or replicate the original findings or propose an alternative exploration. So if you're paying attention to these things, then you'll see new scientific papers popping up all the time saying Planet 9 is not real. So far, every time one of these new papers comes up, the other scientist who and friend of mine who proposed this together, Constantine Batigan, who used to work just down the hall from me when we worked, in real life. Now he works somewhere down the Zoom hall from me. Constantine and I get on the phone and phone will ring and Constantine will say, "Yo, dog, did you read that new paper that just came out?" And literally that's what Constantine would say. And I then go, "Oh my God," when I get these new papers that come out, I break into a cold sweat and I think, we screwed up. There is no Planet 9. I worry about this all the time. So then we retreat into our corners and we scour the paper in detail. And so far, every single time, at least so far, we realized that the paper purporting to show that there's no Planet 9 did something wrong. They never quite wrong, but not exactly right. As in, the paper says, no, these distant objects do not show evidence that they're being pulled by a giant planet and clustered. We realize, your technique actually isn't good enough to detect the clustering that we've already seen, so no. Or the paper says, no, we can explain the orbits with no need for Planet 9. And we then go through carefully and realize, your theory falls apart if you include the gravitational effects of Neptune. So probably try again, or something like this. And then we dutifully explain why this is not right, usually on Twitter, sometimes in all caps. But you know what? From the outside, this looks precisely like what you would do if you were a crank. You would disagree with everything that anybody says it goes against what you believe, and you would dispute every new paper that comes out. Of course, it also looks precisely like what people who will eventually be proven right would do. So which is it? I would say that Planet 9 has some of the key things of theories that will eventually turn out to be correct. So unlike, say, cold fusion or aliens in Venus or aliens on Oumuamua, Planet 9 is some pretty simple physics with some pretty simple predictions. The simple physics is there's a big gravitational, there's a body with a big gravitational field out in the outer solar system, and it does very predictable things that we can tell you ahead of time. Aliens in clouds, alien spaceship, it's not entirely clear, A, how they got there, and B, what other predictions you're gonna make about these things. Cold fusion was just there was nothing specific to predict anyway, so that's less good. Planet 9, we make these predictions and then we find out that they come true. So far, we have shown that as more and more data are collected, the evidence gets better and better. We find new things that Planet 9 predicts that we didn't even think about at the time. Planet 9 seems to work for all those things too. Again, that sounds more like dark energy than it does the other one. And so I would say that there are two bits of good news here. The first is, obviously we're right and everyone else is wrong. I mean, that's just clear. But the second one is, you don't actually have to take my word for it. Planet 9 will be found, and then these arguments will instantly cease. And that's going to be super fun. So the obvious question, when is it going to be found? Where is it going to be found? How come you haven't found it yet? So back when we started this conversation on the TED stage, more than a year ago now, I was pretty optimistic that we were going to find it soon, because I, as I said then, was pretty convinced it was going to be found in some of these old images that already existed, that if we looked through these images, we would see it moving across the sky and find it that way. I'm less optimistic now, mostly because for the past pandemic year, many telescopes were shut down, so we didn't have the opportunity to go to telescopes and search. So instead, we looked through all the old data. We didn't find it. Kind of depressing. I was pretty convinced, as David will attest. I'm like, okay, we can do this TED Talk, but I might find it before we even get up there. I might find it the next day. David, I didn't find it. It's not in those old data. So I think we actually have to go get new data. The good news is, I think it's going to be found now in this newest telescope that's about to come online in a couple of years, the Vera Rubin Observatory being built in Chile. And it's fantastic telescope. It's basically designed to scan the skies night after night after night, looking precisely for things like Planet 9, things that move, things that change, and it's going to see things that are faint. It's going to see things that are far away, and it's going to going to happen. It'll happen within the first year of it going online, which is, I think, 2023, 2024. Telescope is nearing completion, almost ready to go. So for now, I'm 100% convinced it's out there. Looking at the data that we have now, the latest analysis, literally the number we come up with is, it is 1 billion times more likely that you can explain the solar system, outer solar system, with Planet 9 than without Planet 9. And 1 billion, it's a big number. I'll take those odds. So I believe it's going to be found. It's going to be found soon, and that we are in fact not cranks. I'll say that with a question mark. I'm pretty sure we're not cranks. Pretty sure it's out there. Pretty sure we're going to find it. Thanks.
I don't think you're a crank, and I love this.
Might be a crank.
Yes, well, we'll find out. I love this relative ranking of recent scientific hypotheses from questionable to Nobel Prize. That's a good ranking. Well, thank you for that, Mike, and I'll try not to keep my Pluto grudge. But last but not least, we have the amazing Kay Tai. She's a neuroscientist at the Salk Institute, and she gave an amazing talk about controlling brains with light, which I hope you checked out because there's some pretty compelling video in there. But I think she's here to talk about what our brains are like under social isolation. And I have no idea why that might be a pressing topic right now. So take it away, Kay.
Hi, David. It's great to see you and everyone again. Bethany and Mike, it's great to see everybody. And yeah, I wanted to talk about, last time you saw me on the TED stage, I was talking about emotional valence and the neural circuits that we use to determine if something is good or bad. And of course, understanding why it's so important to understand the biology of our minds and our brains, so that we can really unravel the underpinnings of mental health disorders. So today, I'm gonna tell you about what I've been thinking about since then, especially in this pandemic year. So before, my interest in social isolation is long before the pandemic, and we were interested in this for a while. But obviously, I don't need to tell everybody, 2019 versus 2020. This is 2019. This is 2020. It seems worthy to mention that there are a number of consequences, health consequences to social isolation or even perceived loneliness. And so, of course, there are a number of different deficits in mental and physical health, a shorter lifespan. In terms of mental health disorders, there's increased risk for anxiety and depression. In terms of physical health, there's increased morbidity and mortality for both cancer and heart disease, increased inflammation, and increased symptom severity with a viral immune challenge, which seems relevant. So with all of these things together, there's just a really urgent need to understand the neural circuit changes underlying perceived loneliness and social isolation in general to prevent the negative effects. And so many of the most successful species on this planet live in rich social hierarchies, humans included. And one of the questions is, how do individuals maintain their role and know their place within a group? And so this really started with a discovery made by a former postdoc in my lab, Gillian Matthews, who is currently on the job market in the UK. So she is a visionary. And what she discovered was that there are these dorsal raphe nucleus dopamine neurons. This is just a brain region that's best known for actually having serotonin neurons in it, but there are also dopamine neurons mixed in. And after 24 hours of social isolation of a mouse, we can see that there's increased synaptic strength coming onto these dorsal raphe nucleus dopamine neurons, and that these neurons are more active in response to a social target after social isolation than they were after being group housed. So this just suggests that these dorsal raphe nucleus dopamine neurons are sensitive to acute social isolation and can track that state. So if you had a chance to check out my TED Talk, you know that we often use optogenetic tools to control specific projections or specific cell types to see what they can do. And what we found was that when we activated these dorsal raphe nucleus dopamine neurons, animals became more social. There was an increased motivation to seek social contact. But strangely, the stimulation was unpleasant, and animals would if they could avoid the stimulation, they would do so. And so this was confusing at first because, I guess if you tackle the dopamine system in other ways, for example, if you use cocaine, you would be pro-social, but also it was very positively reinforcing, and animals and humans will respond very robustly for dopamine stimulation of other types of dopamine neurons or cocaine. And this was very different. What we saw here was not like anything we'd ever seen with other dopamine neurons. And it had us scratching our head. How could you do something that would make you, both more social and also it's unpleasant? And that was weird. But we likened this to our research in feeding. So from our research looking at feeding behavior, we know that you can eat because something is delicious and you just, you're enjoying this food reward, like a creme brulee after dinner. You don't really need it, but it's just enjoyable. It brings you pleasure and you consume it. Alternatively, you can also be motivated to eat for a negative valence reason. For example, I might dig to the bottom of my backpack for a not very palatable fiber bar or whatever is there, just because I'm so desperate and I want to escape the unpleasant state of being hungry. And similarly, in this analogy to feeding, with social contact, you can be motivated to seek social contact for the social reward. It's just fun to engage and have to socialize. Or you can seek social contact to escape the unpleasant state of being quote unquote lonely. And so loneliness was not a word that had previously been used in neuroscience. It's a word from psychology. And the psychological definition of loneliness includes a number of different criteria, including one, that it increases the motivation to seek social contact. Got that. Two, that it is aversive, it's unpleasant. Got that. Three, that it's distinct from generalized anxiety. That is also true. And then the fourth thing was that it's subjective. And so what I mean by that is, you can be at a party and feel lonely, or you can be alone at home and feel fine and not feel lonely at all. And so this subjective experience is a critical component. How do we get at that in a mouse or even in people? Either way, it's pretty challenging to study quantifiably. But what we were able to look at is the animal social rank. So these are all mice that are raised in the laboratory and have a similar genetic background, but within each cage, each group of mice forms a social hierarchy. And we wildly speculated that perhaps the experience of the social environment was distinct for dominant animals versus subordinate animals. Dominant animals might benefit from, their social environment. Everybody's, they get to mate with whoever they want and prime access to resources. Everybody's engaging in affiliative behavior, which is basically just kind of kissing up. And subordinate animals are denied prime access to resources, denied access to mating, getting beat up by animals that outrank them. And so maybe when subordinate animals are isolated, they feel fine. They feel relieved. The bullies are gone. So this is just a concept to think about how social rank could influence the subjective social experience of an animal. And what we did find, and I almost forgot to mention that when you silence these neurons, we wanted to see what happened if you inhibit these neurons instead of activate them. And if you inhibit them, actually normally when they're group housed, it doesn't change the behavior at all. No change in behavior if they're group housed. But when animals are isolated acutely, essentially all social species, when isolated acutely, like for one day, for example, will show a rebound of social interaction when they are reintroduced to their social group. There will be more interaction once upon reintroduction. And so we found that this post-isolation increase in sociability was also dependent, as was all of these optogenetic manipulations that we did, these three manipulations here, were all dependent on the social rank of the mouse. So the individual animal, whether it was a dominant or subordinate, would change the degree to which each of these manipulations affected behavior. So that suggested to us that maybe we're on the right track. So right now, as a working model, I am sort of thinking of these dorsal raphe nucleus dopamine neurons as an effector system for social homeostasis. And what I'll come to that in a second, but that just means a nucleus that can help to mitigate change and to correct a deficit when there is one. I should also mention that in collaboration with Livia Tomova and Rebecca Sachs at MIT, we were able to see this effect also true in humans. A very similar signal is presented in humans when we show them social stimuli after they've been socially isolated, as we see in humans when they've been food deprived, and then we show them pictures of food. And so here we're only able to socially isolate humans for 10 hours for ethical reasons, since solitary confinement is considered torture. But we were able to see this effect in as short a time as 10 hours. Okay, to this concept of homeostasis. I said this word, what does it mean? Homeostasis comes from the Greek words for same and steady, and it just refers to any process that organisms use to maintain stable conditions for survival. And so in each homeostatic system, you've got different components. You've got a detector system that detects whatever the thing is. So here, there are beautiful examples from many different subfields of neuroscience, looking at energy balance, thermoregulation, osmoregulation, and other homeostatic systems, sleep homeostasis. I'll use right now thermoregulation as an example. So let's say that receptors in my body detect that my body temperature is too high. It's 102 degrees or something. And this gets sent to my control center, which compares to a set point. And if I'm healthy and everything's good, then my set point's probably pretty close to 98.6 degrees. Then, there's a surplus that's detected. This will be fed forward to my effector system, which in the case of thermoregulation might be muscles that move me into the shade or glands that make me start sweating. And then it corrects the change by interacting with the environment. And in this case, the environment is the ambient temperature, pretty straightforward. And then once that's corrected, the whole homeostat shuts off. So needless to say, for social homeostasis, this process is much more complicated. And so a number of different projects in my lab right now are tackling each of these nodes. How do we detect the change? How do we detect the social environment? How do we detect gestures and individuals and know their relative social rank towards ours? How do we store a preferred or desirable amount, a quality or quantity of social contact that is desired, and store that set point and represent it in the brain? And then what are the effector systems that would actually change outputs from the individual to interact with the environment? And then the most challenging part is in stark contrast to thermoregulation, where ambient temperature is essentially the environment. The environment for social homeostasis is the social group, which is, of course, many different individuals with each of their own brains and their own unique mechanisms for processing what to do next. And so this is a non-trivial problem. But the big question that I'm interested in right now is what happens? So why is it that after acute social isolation, almost universally, social species will find that when they are reintroduced to their social group, that they have an increase in motivation to seek social contact. They're performing more affiliative behavior, just engaging in a pro-social manner, more social engagement. However, with chronic social isolation in humans, monkeys, rodents, even fruit flies, what we get instead is that when they are reintroduced to their social group, their previous optimum, they now display antisocial behaviors, increased territorial behavior, increased aggressive behavior, which is not always antisocial, but in the case of territorial behavior, it is more avoidant behavior. And so how do we go from something that's a positive valence response to a negative valence response when the manipulation, social isolation, and the stimulus, reintroduction to the social group, are the same? What gives? And so this is something that, in the past the past year I've thought about a lot. And so if we put on the y-axis here, quality and or quantity of social contact, and this is a conceptual model that we are in the process of formalizing into a mathematical model. Upon an event like social isolation, the effector system will be activated. The animal will increase motivation to seek social contact, and then this deficit can be rectified. But sometimes it can't be rectified. Social isolation will occur, effector system will be activated, but for whatever reason, returning to the previous optimum is not possible. Then after a certain amount of time, or perhaps it's not time, it's correction efforts made that have been failed or rejected. At some point, we propose that there's a set point adaptation, that the homeostatic set point is adjusted, and this can be seen by the reduction of efforts being made. And then this kind of becomes the new normal. You adapt to what you're getting. And then this could explain why, when we are reintroduced to our previous optimum, reintroduced to our social group, that after chronic social isolation, this can actually be not optimal. This can seem like a surplus. And many of the behaviors we see with chronic isolated individuals being reintroduced to the social group looks more like overcrowding than it does like their previous optimum. And so this is just one way to think about how we could be getting to this phenomenon. And so this is something that we're working on in the lab. And I want to thank everyone in my lab who's contributed to all these projects, our collaborators, our funding sources, and you for your attention.
Not at all socially isolated. It takes a team to deliver the science. So, and you haven't made me anxious at all about this pandemic experience. Well, thanks to all of our speakers. I hope you can rejoin me. I am conscious that we have eaten up 99% of the time. However, I'm going to ask a rapid fire question of each of you, and I want you to kind of answer it in a sentence or two. And we'll start with you, Bethany. This question came in from Uma Ramachandran. I hope I pronounced that close. Any role for oxylipins in human health or medicine?
Absolutely. They're involved in lots of different pathways in response to wounding, viruses, including COVID-19 potentially, because it affects the lungs, diabetes, bacterial infection. Check out my TED Talk if you're interested in it. It's not what I study, but there's a wealth of literature on it.
Great job. Also points for brevity of answer. Moving on to UK, Joe Bridger wants to know, how do brains react in isolation tanks? Do we know what happens when you kind of sensory deprive yourself?
That's a great question. I think there are some adaptations, and you go into a different state, an internal state. So some people might call that a network state, but you go into a different brain state when you are deprived of sensory input.
Great. That didn't freak me out at all either. And Mike, last but not least, Andrea wants to know, could AI help with this search for Planet 9, given everything you have to search?
Yeah, so it already is. For most of what we do, rather than staring at images ourselves, we train computer systems to help us identify stuff and put things together. So we really have come a long ways since the days of Clyde Tombaugh finding Pluto by manually looking at two big pieces of glass. This is quite a big change.
This is good. This is good. And you guys did so good that I think I can sneak in a question for all of you.
Can I sneak in a question?
No. Oh, for somebody, only if it's for somebody else.
Well, it's for Kay. Because you talked all about how all of us have now had our set points lowered and people freak us out. Yes, I agree. Do you think those set points will reset to where they were in the past, given previous levels of engagement?
I think that the set points are adaptable in both directions. If you were to engage in overcrowding, you would adapt to that as well. So I think we have no reason to believe that our brains are not symmetrically plastic. So I feel hopeful.
There's hope.
Science summer camp.
There's hope for us. I hope not to be too weird once we get back in person, but I was pretty weird before all this, so it's okay. Well, I think we are at time, so I want to thank all three of you for those excellent and engaging presentations, and I'm pretty sure that this spawned a lot more questions than answers in our audience. So I look forward to continuing the conversation, and I will turn it back over to Anne and Rick.
Oh, Anne, you're unmuted.
Muted. I am muted. It's a rookie mistake. What am I
We made it so
doing?
far.
Well, of course, I have to say all over again how much we appreciate all of you. We couldn't do these events without amazing presenters, and we were really glad to pluck the three of you off of our TED at NAS stage. And of course, we feel really especially happy that we got you, David, because it wouldn't feel like a real TED reunion unless we had you. I think all three of our presenters would have felt like, what? No, David? So we're really glad that we got the band back together, and we appreciate our audience being here, and we thank you once again for sticking with us. And we'll be back in your inbox next week because that's what we do. And we will be back with actually an alumni from the TED stage. I think multiple times on the TED stage, because David Gallo will be joining us next week. He's one of our dear friends and wonderful science colleagues. He's I think he calls himself an oceanographic bon vivant. So tune in next week for Dave Gallo, because he's going to be awesome.
I just call him a science ninja. I don't know.
Well,
Bon vivant. No. Anyway.
totally works. Well, he tries to pretend he's sophisticated. We know it's not true.
Well, thanks everybody for this week, and we'll see you next week.
Bye.